It is now my distinct pleasure to introduce Team 3 representing Georgia State. Team 3 consisting of Amy Sims, Alex Mark, Bo Martin, and Aditya Natham, who will be presenting on hyperparasites, nature's equilibrator. Please take it away, Team 3.
Thank you, Michael. Our presentation is called Nature's Equilibrator, Hyperparasites. The four of us, Aditya, Alex, Bo, and me, Amy, represent Team 3 from Georgia State University. So naturalists observe a flea has smaller fleas that on him prey, and these have smaller fleas to bite him, and so proceeds ad infinitum. Swift was describing the tendency of poets to feed off the work of earlier poets. What Swift could not have known in 1733 was that he was describing a product of evolution that would have profound effects on the ecosystems of the world. You may have heard of the parasitic stunt nematode, named for its propensity to deform the roots of field and vegetable crops, destroying billions of dollars of vegetation per year. What you may not know is that the stunt nematode has its own parasite. The hyperparasite, Dactylella coinopaga. This diagram depicts Dactylella spindling elongated cells, entangling a stunt nematode in its noose structure, where it traps it and then absorbs its nutrients through filaments. Hyperparasitism evolved from primary parasitism, a system that began before the Mesozoic era, over 250 million years ago. Parasites are dependent, ruthless, and efficient. So efficient, in fact, that it has evolved along separate lineages several hundred times. Transitions from a free-living existence to a parasitic one probably outnumber any other type of major evolutionary shift in life history strategy. As life diversified and complexified along evolutionary lines, organisms began to have complex interactions with one another. These interactions generated new niches for individuals in biosystems. Mother Nature had selected parasitism to solve the problem of overpopulation, but she soon saw that the solution to one problem created another. The system was not sustainable. She needed a way to control her hungry, demanding child before it killed every organism on Earth, including itself. We discovered that most pathogens are likely to host parasites of their own, that is, hyperparasites. And so we wondered, why do hyperparasites consistently emerge in biosystems, despite occupying such a narrow niche? Necessity is the mother of invention, and the great inventor, Mother Nature herself, found a need and filled it. Enter the parasite of other parasites, the hyperparasite. Some hyperparasites hitchhike with their parasite hosts. Those hosts are not as virulent as other parasites, and the system trends toward hypermutualism. Because that system is mutualistic, the primary parasite is not destroyed, and it continues to effect relatively mild destruction. But for the truly virulent primary parasites, nature doesn't send in a hitchhiker, she sends in the repo man. These deadly hyperparasites commandeer their host's faculties and use them for their own propagation, killing the host in the process. But luckily for the primary host, this violence inhibits the spread of the primary parasite, which enables the primary host to maintain its population. Which leads us to our hypothesis that hyperparasites consistently emerge in nature in order to equilibrate parasitic systems that would otherwise be crushed under the weight of the highly efficient primary parasite. Hyperparasitism plays a critical role in the evolution and epidemiology of microbial pathogens, a role designed, curated, and championed by Mother Nature herself. Let's look at some examples. The fungus C. parasitica infects chestnut trees with blight, causing significant destruction. The CHV1 hyperparasite preys on the primary parasite and thwarts its destructive path. This photograph depicts the hyperparasite Ampelomyces quisqualis, which are intracellular fungal mycoparasites that grow inside the mycelia of their hosts. They prey on the fungal pathogens called P. plantagenus, which is a powdery mildew that parasitizes the plant P. lanceolata, used in herbal teas and remedies. Now that we've explored some general examples of hyperparasites, Alex will introduce us to the specific system that is the topic of our research.
Thank you, Amy. So to model our question, we studied the relationship dynamics between carpenter ants, the zombie ant fungus, and their hyperparasite. The ants, or Camponotus leonardi, are commonly found in warm tropical climates like Brazil or Thailand, and they nest in the tree canopies of rainforests where their populations can grow up into the millions. The ants spend most of their time in the canopy, but foragers do come down to the forest floor, and that is where they pick up the fungus. So Ophiocordyceps unilateralis, or the zombie ant fungus, is both an obligate parasite and a parasite to the carpenter ants, which means that it needs the ant to complete its life cycle while also ending the ant's life cycle. And so here on the bottom left, you can see a healthy carpenter ant, and then the next two pictures are of dead ants with a mushroom coming out of their thorax. Next slide, please. So in this slide, you can see a forager ant picking up the fungus, which then penetrates the ant's exoskeleton. It surrounds their muscular system and their brain, but interestingly, isn't actually inside any of these muscles. And so this zombie ant no longer functions regularly as an ant would, but rather exhibits the traits of a fungus in ant's clothing. And so to ensure sporulation or reproduction, the fungus essentially uses the ant as a transportation unit. Because the ant lives up in the tree canopy, the fungus will make the ant experience these full body convulsions to ensure that the ant either falls to the ground or remains on the ground if it's already down there. Once it's at ground level, the fungus searches for a suitable environment to produce spores. And after it's found a good spot for reproduction, the infected ant will latch its mandibles into a nearby leaf and die. After death, the ant's mandibular muscles will atrophy, resulting in lockjaw behavior, which keeps the dead ant anchored to the leaf. And so left unchecked, the zombie ant fungus can devastate ant populations. Luckily, nature has created a way to suppress these fungi. Next slide, please. So this introduces the third actor in our system, the hyperparasite. So the hyperparasite is also an obligate parasite to the fungus and will inhibit fungal sporulation by essentially castrating the fungus during its maturation period, which lasts about a month after the mushroom begins to sprout. And so more than half the dead ants in each graveyard harbor hyperparasites, and graveyards are just aggregations of dead infected ants. Next slide, please. So this picture shows the ants' habitat. As I mentioned earlier, the ants nest up in the tree canopies, and the nests are represented by these orange symbols in the trees. And to get through the canopy, they follow these black lines, which represent aerial trails. So this line coming down the tree is the path the foragers will take to the ground, and this is where they'll pick up the fungus. They'll have an infection period of about 4 to 10 days. So the same day they pick it up, they can immediately return to their colony. But fortunately, healthy ants can't get infected by this patient zero because the spores are too large for that kind of transmission. So if the infected ant is still in the tree at this point, the fungus will basically shake it out, like I mentioned earlier. It will drop to the ground, where later it can be found in a graveyard represented by these green circles. Next slide, please. So this diagram basically shows the entire process. The forager ant will descend from the canopy, pick up the fungus, and find a suitable environment for fungal reproduction. 3 to 6 days after host death, stroma, the spore-bearing structure, will emerge from the ant's thorax, and it will take some time for the fungus to actually sporulate. So during that period, it's vulnerable to being hyperparasitized. But if it manages to get past that maturation period, it will produce spores. And in terms of our actual model, which Bo will explain more in depth in a second, there are no classes of mature or immature fungi because that isn't relevant to the question that we wanted our model to answer. And there are also no graveyards for the same reasons. Bo.
Thank you, Alex. So like the other groups, we chose to use NetLogo, which is an agent-based modeling system. So first, I'll just go ahead and explain some of the main mechanisms of our model. So in the picture that you're seeing here, the red ants are ants that have been infected with the fungus. So in the model, there's the ants and there's the patches. And so if a patch is gray, that means it has fungus. And if a patch is white, that means it has the hyperparasite. And so to model the way the ants interact with the fungus in real life, so the patches of fungus randomly generate, and if the ants walk over them, they become infected, which are the red ants. And then so normally the ants would climb up the tree to their 25 centimeter leaf, And then like a couple of days later would either drop their spores or get the hyperparasite. So in this case, the ants pick up the infection from one patch, removing it from that patch, and then they go to another patch. And if the patch they're on when they die doesn't have fungus or hyperparasite, they'll transmit the fungus there. And if it does have the hyperparasite, they'll actually transmit the hyperparasite. And so originally in the model, we had the hyperparasite didn't belong to the patches. It was just whenever the ants died, they had a chance to either transmit the fungus or not transmit the fungus. Because like Alex said, the hyperparasite essentially castrates the fungus, preventing it from reproducing. But this didn't allow us to model the fact that the hyperparasites are not static and there's actually like a hyperparasite population. So in this case, with this spread, the model now encompasses the fact that like there's a hyperparasite population and a fungus population sort of competing against each other. Next slide, please. So in this picture right here is just one run of the model about halfway through. And so now I'll talk about some of the assumptions and simplifications of our model. So one of the big ones is that the ants move randomly and that we also only model it in 2 dimensions. Because again, like Alex showed earlier, the carpenter ants actually like live in trees and the ants are coming down to forage is where they can pick up the fungus. And also like, you know, when you see ants normally, like they're moving in lines and stuff. They're not just like all running around all over the place. So, but we weren't able to encompass that in our model. And also that the ants always, so each patch that has a fungus, the ants will always get the fungus from that patch if they walk over it. Whereas in real life, there's a chance that they just might not pick up the fungus. Next slide, please. So on the top slide, you can see the model being run without the hyperparasite, and on the bottom is the model being run with the hyperparasite. So of course, our main question was, why do hyperparasites emerge in biosystems? And like Amy said in the beginning, without the hyperparasites, the fungus, as you can see, quickly decimate the ant population, but then also they die. So the hyperparasite, once the hyperparasite is introduced, as you can see in the bottom model, it leads to a more stabilized system, which was predicted by our ODEs and compartmental model that Amy is now going to introduce you to.
Thank you, Bo. We chose a compartment model because it allowed us to track our ants through their cycles, and because it aided us in writing the ordinary differential equations we needed in order to find the ratios of endemic equilibrium. In this model, S represents the susceptible ant population, I the infected ant population, F is the portion of infected ants that sporulate the fruiting bodies of the fungus, and H is the portion of the infected ants that are covered by the hyperparasite, meaning they do not spread the fungus. Theta is the rate at which the queen ant feeds her offspring into the susceptible compartment. If an ant is infected by cordyceps, it moves to the infected compartment I. From there, the path branches off according to little f, a sigmoid function that represents the fraction of the infected ants that go to the hyperparasite compartment. We chose the sigmoid function because it is steep where h is small and flattens out as h grows, asymptoting to a value of 1. It was chosen to represent how quickly the hyperparasite spreads and how well it helps maintain a sizable chunk of ants that can resist infection. The sigmoid function, little f, ranges from 0 to 1 because its value is a fraction of the whole. Some infected ants go to h, the others go to f. The rate of change of the susceptible ant population equals positive theta, new births from the queen, minus the per capita infection rate beta times the population of available ants and available cordyceps fungus. The infected compartment receives all of the infected ants from S, so that term is equal and negated, All infected ants die, so they leave I after a certain number of days at a rate of rho, which is always equal to 1 because of the 100% death rate, times its population. The positive rates of change for F and H equal what they get from I minus their per capita rate of decay, tau and omega, respectively. The function shown here is F of H, the percentage of infected ants that are covered by the hyperparasite. We customize the sigmoid function to yield a value of 0 when the hyperparasite presence is 0, and to asymptote to 1 when the hyperparasite presence eclipses the cordyceps population. A represents the population of hyperparasite that yields a 50% conversion. And we saw that by adjusting the value for A, we can steepen or flatten the curve. We then took to Excel to find the value for A that generates a stable equilibrium for the ant cordyceps hyperparasite ecosystem. Setting A to 20 makes the hyperparasite peak too early. The fungus population crashes and the ant population swirls out of control. When A is too high, here at 50, the hyperparasite peaks too late. The ant population crashes and the hyperparasite is ineffective at stabilizing the system. We found that a value of 30 for A yields a stable equilibrium, keeping in mind that these are merely ratios and do not represent actual values of populations. Notice that the blue line, which represents the susceptible ant population, plummets early, inversely proportional to the red line infected population. The green line fungus population peaks after the red line because of the time delay between the infection of the ant and the sporulation of the fungus. The unit time here is 1 year, and we notice that around the 3-month mark, the fungal population becomes constant and the other 3 populations begin to asymptote together nicely. Aditya?
So all of this into consideration, here is our conclusion and questions we're still thinking about. So from killing ants to trees, parasites destroy and create disproportionate ecosystems. So as a result, hyperparasites came into existence. But why do they consistently emerge in biosystems? We dived into the idea of hyperparasites through the relationship between the carpenter ants, the parasites, and the hyperparasites. We created a compartment model alongside a system of differential equations to try and help explain our observations and research and come to a conclusion that hyperparasites can stabilize the ecosystem. To visualize the effects of certain parameters set in the compartment model in ODEs, we proceeded with the agent-based modeling, which is NetLogo. There we could see the impact hyperparasites made to the ant and fungi ecosystem. Like Bo mentioned, without hyperparasites, the model demonstrated that the ant population gets out of hand and the fungi died out, while with the hyperparasite, the model showed that the ant and fungi stabilized. Understanding the graphs and models led us to believe the importance of hyperparasites and why they're prevalent throughout many ecosystems, as hyperparasites stabilize host and parasite relationships. Having a stabilized system has many benefits and potential applications, such as for farming or having a self-sustaining ecosystem. This project has led us to think about other questions like, what if the function of the ants are proportionate to the density of the trees? It raises ecological questions, like does deforestation play a role in the host and parasitic relationship? And does geography impact the effectiveness of hyperparasites? Other research we looked at included accurate ant data, and we could potentially look at 3D modeling, adding the vertical element trees too. And the top picture is a terrarium, which is a self-sustaining plant ecosystem, and the bottom is a scientist farming cordyceps. This type has herbal remedies for humans, such as anti-tumor effects and anti-aging effects. It's called Cordyceps militaris.
Penultimately, our heartfelt thanks to our brilliant and long-suffering benevolent overlords, Michael Levine and Maya Mims, for an unforgettable 6 weeks of fun and learning.
Thank you.
Thank you.
And finally, certainly, last but certainly not least, so many citations.
Applause to Team 3. Thank you all for a wonderful talk. And if you're having trouble forgetting about it, then I'm sure there's a pill for that. We
All
have
right.
5 minutes for questions for team 3.
All right. I just want to say that was great. Y'all popped off in the beginning. I was not ready for that. Like Amy, you just like you were feeling yourself. And
I was.
like, and what was it? What Alex, you said that line where you're talking about, what is it? A fungus in ants' clothing. Like, I knew that's what it was, but like it didn't hit me. This is literally Invasion of the Body Snatchers. This is what we're dealing with right now. And so like just everything, I loved it. It was great. And I really liked how this is kind of like the sister project, I guess, to ours, because in a way your project kind of inspired ours, because hypertumor, that originates from hyperparasite, like what are the chances, right? And so yeah, this is great. I liked it.
Thank you, Cameron.
I'm actually really interested in looking back at some of your ODE models and kind of talking about where that last equation that you went and graphed from, if you mind going back.
This one?
Yes, the sigmoid function. Do you mind talking a little bit about that? What you kind of use, like what initial function you used and tweaked to get your results? So
the original function that we used was the sort of generic sigmoid function that was it, as opposed to what you see on the screen there, it was 1/1 plus e to the negative x. That gave us a y-intercept of 0.5. So we just changed the y-intercept. Then we needed, but then it only asymptote to 0.5. So we multiplied it by 2. Then we realized that the graph peaked too early. The first one was this red in the middle. And we realized that if it peaked too early, that the hyperparasite would take over too quickly. We needed to flatten that curve a little bit. So we decided to make the exponent for E to be a function of the presence of the hyperparasite. And A is going to be the population of the hyperparasite where the hyperparasite yields a 50% conversion. So we were trying to get that to where it was kind of the halfway point. Obviously, because it's a sigmoid curve, it's going to be steep at the beginning and then flatten out. So that's when we took to Excel to find the value of A that would yield that stable equilibrium.
Yeah, let me piggyback and say like that. In principle, you could think of a really big compartment model that takes, okay, there's susceptible ants become infected with cordyceps. The cordyceps then have to fruit, and then the fruited cordyceps has to contact with a hyperparasite spore. Then that has to fruit. You could construct a very long daisy chain of compartments, but this piece of modeling was very cleverly designed to sort of say, can we reduce that dimensionality in a way that just sort of summarizes that some of the infected ants will sporulate cordyceps spores, some of them will sporulate hyperparasite spores. And the idea was that, well, just some fraction will become hyperparasite. What fraction? Well, a fraction that gets closer to 1, the more hyperparasite there is. Then I think Amy did a really good job of then telling the story of, okay, but this shape parameter A is determining how effective, right, how aggressive that hyperparasite is. And indeed, there's again this sort of Goldilocks zone that I think is a story that's emerged in a lot of these biosystems, which is really compelling.
Thank you, Michael.
We have another minute or so for questions.
I can jump in with another question. What are some ways that you think of possibly expanding on the system and possibly creating a more like full-scale model?
Yeah, we really were limited by 2 dimensions in the NetLogo model because the ants live up in the tree canopies of rainforests in tropical regions. And so we were unable to keep them off the ground. And we definitely wanted to, thought about adding that into the model, but then that added, significant difficulty to the model. We really wanted to pare it down, as I'm sure everybody in the group has experienced having to pare down. We would also like to explore the function of the population of ants as a function of the density of the trees, because since they live in the trees, if the tree density is lower, then the ant population is going to go down, and ants are vital to life on Earth. I'm sure that everyone is aware, but they contribute to aerating and properly draining the soil, changing the soil chemistry. They even plant seeds, and it's been hypothesized by people smarter than me that we could not survive without ants. So it's the destruction of the rainforest and all over the world is something that's definitely affects the ant population and what they do for us.